and validation of tests for SARS-CoV-2. Please remember that during this town hall, we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene, and thanks everyone for joining us again this week. I have a couple of updates this afternoon. First, as you may have seen, on Monday, we issued a new guidance document for the policy for evaluating impact of viral mutations on COVID-19 tests. Obviously, this is a topic that we've talked about here on the town hall quite a bit recently, and we now have this guidance document out that was put out along with a suite of guidance documents from the other centers as well, all on the same topic for different medical products. So our guidance provides information on evaluating the potential impact of emerging and future viral genetic mutations on COVID-19 tests, including design considerations and ongoing monitoring. As you all know, we've already issued a safety alert about this topic and identified a few tests that are known to be impacted, though at this time that impact does not appear to be significant. We continue to monitor and will put out additional alerts as needed if and when future impacted tests are identified. The guidance goes into the monitoring that FDA does, as well as describing recommendations for test developers, such as considering the potential for future viral genetic mutations when designing your test, conducting your own routine monitoring to evaluate the potential impact of new and emerging genetic mutations, and information that we would expect to be provided to FDA, both in an EUA request and in supplemental EUA requests or reports after authorization with ongoing monitoring. And it does provide more detailed recommendations for developers of molecular SARS-CoV-2 tests, and also discusses the sort of landscape of monitoring for antigen and serology SARS-CoV-2 tests, and forecasts that we will put additional information regarding that monitoring in the EUA templates as more details are worked out. So that guidance can be found on our website, and if there are any questions, we're happy to take them on the call today, or you can also send questions to the mailbox, as you're all familiar with. The other topic that I wanted to bring up before we get into the questions is to clarify some comments that were made last week regarding usability and user comprehension studies. One of the risks of a town hall forum like this is that Tim and I don't always have the source documents open in front of us, so we don't always have all of the details to best answer the questions that come up. So sometimes we like to do a little research and come back and clarify for everyone. So we had a question last week about questionnaires following usability studies, and there may have been some confusion between usability studies and user comprehension studies. So we want to clarify that those are two different things that serve different purposes and provide different validation information. Usability studies assess whether the intended user of the device can use the device properly without serious use errors or problems. And this testing typically would include an observational assessment of the users performing the testing, an assessment of the sample collected or the testing performed by the intended user, and an interview or questionnaire to assess the user's perspectives on the usability of the device. Our current template for molecular and antigen diagnostic tests for tests outside of healthcare settings, so I think we refer to it as the non-lab or the home use template, does include a recommendation to assess the user's perspective on the usability of the device using a questionnaire, consistent with the recommendations in our 2016 human factors guidance. I will note that the question last week referred to that 2016 human factors guidance. And I do want to point out that some of the details in that guidance, while they are the general concepts are applicable to IVDs, there are specific details that are necessary to consider for IVDs, since the guidance document is written for evaluating user interfaces, which is different from evaluating an IVD where that involves collecting a sample and performing a test, as opposed to simply interfacing with a user interface. So those are usability studies. And then separate from usability studies, there are user comprehension studies. Excuse me. User comprehension studies are intended to objectively assess the intended user's understanding or comprehension of critical elements and concepts in the labeling. These studies are particularly important for tests that are intended for use by lay users that do not include the involvement of a healthcare provider. The current template for non-lab tests does not discuss user comprehension studies, but we do think that this is very important to assess, especially for tests where there's not a healthcare provider involvement, to consider user comprehension and assess the comprehension of labeling for devices where the results are returned to the lay user. And I want to note, particularly for serology tests, and we are currently working on a template to provide recommendations for validation for serology tests performed outside of healthcare settings. There are significant risks associated with lay user misinterpretation and misuse of serology tests, since they should not be used to diagnose acute infection, and it's unknown whether antibodies confer immunity or protection from infection, and if so, how long that may last. The clinical significance of serology test results, especially in individuals that have received vaccine, is unknown. So given the current limitations of serology testing, lay users may misunderstand and potentially misuse serology test results. So it's particularly important to assess the user's understanding of the test, including its limitations. So with that clarification of that discussion from last week, that concludes my updates, and I don't think Tim has any specific updates this week.
Yeah, so, Toby,
yeah.
Can you hear me? Yeah, I just wanted to update one thing. I just want to reemphasize our current priorities for review at the FDA for tests. So obviously, home over-the-counter tests, including rapid antigen tests, are a high priority, as well as home prescription tests, including rapid antigen tests. Also, point-of-care tests, including rapid antigen tests, as well as extremely high-throughput central lab tests, pretty much of any sort. We're really looking for both molecular and antigen, the diagnostic tests for the high-throughput systems. So with that, Toby and operator, I think we can go to our questions, the question period. Thank you.
Thank you. At this time, if you would like to ask a question, please press *1 on your touch-tone phone. Please ensure that your line is unmuted, and please record your name when prompted to be introduced. Once again, it is *1 at this time. Our first question is from Ariana Perez. Your line is open.
Hi, my name is Ariana with UserWise. I have a question. Is an anterior nasal swab with no stopper allowable for use with children under 14 in an OTC context, or does the FDA have data suggesting that nasal swabs with no stopper are unsafe for children under 14, even for anterior nasal swabs?
So we're interested in safety in the home environment, and especially when untrained non-healthcare people are doing swabbing at home. So for an anterior nasal swab, if clearly shown and in the study show that there's no harm done, that's good. If you're going to go further in than an anterior nasal swab, such as a mid-turbinate swab, that's at the point that we would like some sort of safety features. And certainly, we encourage developers to, even for anterior nasal swabs, think about safety in that situation.
Okay, so for anterior nasal swabs, it's acceptable to not have a stopper, but FDA is requesting stoppers for mid-turbinate swabs.
A stopper would be ideal. There are potential other medications. There are different sort of swabs with, some sort of adapter that some developers have used. But something, to make sure that the children are safe when they're getting swabbed.
Okay, thank you.
Thank you. Our next question is from Didi Datra. Your line is open.
Hi. My question is related to the new guidance that was issued about the impact of viral mutations. So the first part of my question is, can you please recommend a source for obtaining specimens positive for these mutations? And the second part is there any guidance that we can get on how to evaluate the performance of the test? So because we are developing a serology test for point of care use, and we're intending to submit the EUA pretty soon.
So for serology tests, there's current thinking is there's some things that we can do. For example, every serology developer would know the epitope of the virus that's being used. They know the sequence of amino acids. So you can check the known public databases for any prevalent mutations that impact the amino acid sequence of your epitope in your assay. Only if it does impact directly your epitope, or if you have other means of assessing whether amino acid change outside the epitope could impact things. But I think, right now, the best way to go about this is to look at that epitope antigen that you're using in your assay and make sure there aren't any prevalent. And we usually use the 5% rule, but check out the guidance. 5% or below, or 5% in total or below, or it doesn't push the performance of the assay down below what we would usually expect to see for authorization. And obviously, if you do have an epitope that has a prevalent mutation that leads to an amino acid change, you want to assess what the potential impact on that amino acid change is. Is it something? Is it something that shouldn't actually change too much? It may be a very similar new amino acid, but in all likelihood, if it's changing an amino acid, it may be due to immune function that may allow it to somewhat evade the patient's immune system. And so then how do you know beyond that for serology test if it impacts? And that would probably involve trying to find immune serum or plasma from the patient that carries that mutation or variant in order to assess that. But again, it's all risk-based. So if that mutation or variant is less prevalent right now, we may just authorize the test with labeling that says there's a potential, problem with this mutation and with this assay. So hopefully that's helpful enough. And we hope, that immune serum will be available to developers. And, we're certainly thinking about all of those things. So, but I don't have any program to announce right now, other than what I've just talked about as far as looking at amino acid changes in the serology epitope molecule.
Okay, thank you. But just to clarify, so can FDA recommend a source where we can obtain specimens that may be positive for the most prevalent mutations right now?
Yeah, it would be in the case of serology tests, it would be immune plasma or serum from a patient known to have been exposed by the virus that could genotype that could impact your assay. So that's obviously going to be a challenging sample for anybody to accumulate. So we're certainly in the process of looking into that, but I have no solution. I have no recommendation at this point, unfortunately.
Okay, thank you so much.
Thank you. Your next question is from Vanna Smith. Your line is open.
Thank you. Tim, first, thanks to all the reviewers for all their hard work. And I want to go back to January when you did an update on the MDUFA hold or backlog, since many of us are interested in both MDUFA submissions and EUA. I was wondering if you could give an update.
So just for those that don't know, because of the volume of COVID applications, all applications in total were for COVID, including pre-EUA, original EUAs, EUA supplements, amendments. We're seeing somewhere between 200 and 300 new applications a month still. And that's just a tremendous volume, but obviously it has great import for the country. And as a result, we did move headcount from non-COVID work to COVID work, and in the process took people off of regular MDUFA non-COVID work. And as a result, we, because of the workload volume, we did put files on hold. And those holds, I was going to say, are not indefinite. They will come to a very specific end, no matter what file. So all files will be treated equally. This is not an indefinite hold, and we will be communicating that with each sponsor. So if we can't get back to it on the first hold, the next communication will spell out exactly when we can get back to that file, and so it will not go on indefinitely. Hopefully that addressed your question.
Yes, thank you.
Mm-hmm.
Thank you. Our next question is from Joanne Gonzalez. Your line is open.
Thank you for taking my question. Has the agency authorized any mobile medical applications, either for COVID-19 or otherwise, that are supported on both iOS and Android? And if so, were there any specific requirements asked of the test developers to show equivalence between the two platforms? Or if there are any specific areas of concern to support a system on both platforms? What we've seen is there may be significant differences in the code base in order to produce equivalent performance. Do you have any thoughts on what studies are required to support their equivalency?
So the test is a serology test or an antigen test?
It's a rapid antigen test using a mobile medical app.
Okay, and that app will read the results of the test, or will it just be an app for reporting function, not involved with reading the results of the test? Or
it will involve. It will provide step-by-step instructions, and then it will also capture the image and provide results, and will have the function for reporting also.
So it's capturing the image, and is it based on that image? Is it making the assessment of whether it's positive or negative or indeterminate?
Correct.
Okay. So that obviously cell phones are different, even within a family, iOS or Android. The different models that are available have different cameras, different resolutions. Lighting may play a big role, exactly how you hold something, and that could have differences between even within a family of cell phones or smartphones. So it's best if there is studies showing equivalency between those. They don't have to be on actual patient samples. So you could validate with, say, with one model, or you could validate with any of the models that you would accept in the study of home users, but then show basically with analytical work that there's with high, medium, and low positive samples in different lighting conditions, that the different models perform equivalently, and which ones don't. So there's a great deal of flexibility here, and so the best thing you do is to propose a study, design, however you want to run your study, and then how you would propose to validate the different models that you would allow in the authorization. But we do think that in the labeling, the models that you've validated should be stated, and that obviously performance outside of that would not be known.
Okay, thank you very much.
Mm-hmm.
Thank you. Our next question is from Roxanne Chan. Your line is open.
Thank you for taking my call. Regarding a multi-analyte molecular test validation with clinical samples, we understand the need of using an FDA-cleared comparator assays for flu A and B. Does FDA have any guidance on, say, how recent these FDA-cleared comparator assays we should use? And also, given the flu rate is so low, we only have access to archived flu samples. Does FDA have any guidance on the age and the storage conditions of these archived samples to be applicable in a clinical validation?
Okay, please stay on the line because I want to make sure I've unpacked all of that question, because there's quite a few. We'll talk about bank samples. You can use bank samples. We do ask that you support, through stability studies, the use of say, the oldest of those samples. But there's no problem using bank samples for the non-SARS target. There will be a post-market commitment to do a prospective study for those analytes, but we understand that especially flu, there's very little flu circulating, so bank sample is really the only way that you're going to be able to assess that. I'm not aware of any limitations on the authorized comparator tests for non-SARS targets, as long as it's FDA authorized molecular test, that would be acceptable to us.
Fantastic. Thank you very much.
Thank you. Our next question is from Ron Domingo. Your line is open, sir.
Thanks, Tim and Toby. My question has two scenarios. So what is the agency's view on software changes that improve performance in the context of the pandemic? So in this one scenario, after authorization, the developer finds a way to improve the algorithm, and it does increase the PPA from 90% to 92% without any changes in usability. Based on the guidance, FDA guidance, deciding when to submit a 510K for a software change to an existing device, this would require a new 510K since it has a direct impact on diagnostic performance. Would a similar rationale apply here to an EUA? So would we require a new EUA or EUA amendment?
So it depends. So we are encouraging developers, if they know that they're going to make software modifications that may alter the performance and hopefully improve performance. Or you just don't know if it's going to alter the performance, but you're planning on doing it. During the EUA process that you secure from the FDA sort of pre-approval of pre-authorization of a change, a modification, which would include covering the kinds of changes you would make and how you would assess performance and what would represent inadequate performance, acceptable after you evaluate that change. And if that is built into your authorization, then if everything looks great and it meets everything that the FDA and the developer expected to see, there would be no need to come in for a modified change unless there would be an updated labeling, any different instructions, or if you wanted to actually show that you have better performance and modify the data in your EUA authorization to show that your test is now more sensitive. If you already have an EUA authorization and you had not preset that that change modification procedure, then we'd want to see the data as an amendment. If this is a point of care or a home test, we would need to see that data and review it and reauthorize prior to you launching that. However, if you have a kit for use in high complexity labs, you can validate that, submit the validation to us, and immediately launch that change while we review it and prior to making the EUA authorization decision.
Okay, yeah, thank you. I was going to ask the second scenario, but your response was very thorough. Thank you, Tim.
You're welcome, thanks.
Thank you. Our next question is from Karen Gerard. Your line is open.
Hi, thank you for taking my call. Earlier in the pandemic, the FDA allowed for enrichment due to low prevalence, but not long ago we had peak prevalence that allowed for very quick enrollment of both symptomatic and asymptomatic subjects in prospective clinical trials. The current rates are dropping, though. So my question is specifically, for molecular tests, what does FDA think about recruiting subjects that have had a positive antigen or PCR test in the past 48 hours?
This is something probably we want to take a look at prior to you starting the study if you don't want to have the study be at risk. But definitely, I have publicly stated for accumulating multiple times, I think, on this town hall call and other settings as well, that for enriching for asymptomatic individuals, you can connect with some sort of regular testing program. And if they're positive, then you can resample and use it in your test. We would just want to know that design. When you do that, we also, of course, want to see that you keep everybody blinded to the prior results and include at least an equal number of negative results in that blinded retesting. So if somebody gets the test, they're negative, then we would want a portion of them included, as well as a portion, that you need to get to the recommended number of positives for the study.
Okay, thank you very much.
Thank you. Our next question is from Kadumi Venkat. Your line is open, sir.
Thank you for taking the call. I am Kodumudi Venkateswaran from Tetracore. I wanted to ask about the serology test, and especially now the vaccinations are there, the differentiation of vaccination versus infection through serology. I think the last meeting you mentioned that probably if such a test comes there, that's a higher priority. earlier when you were mentioning that was not discussed. So can you please elaborate still if a serology test that comes to differentiate vaccination versus infection, is it of interest to the agency? Thank you.
Yes. If the application is complete and has evidence that could support such a claim, then we will make that review a high priority. The study designs for supporting such use, though, are not insignificant, and so just look at that. And you probably want to run a SOP, a study design SOP by the FDA, to see if that is going to support whatever claim that you may seek.
Thank you.
Thank you. Our next question is from Annie Bell. Your line is open.
Hi there. Can you hear me? Hello?
Yes.
Okay, great.
Yes.
Thanks.
So
I have a two-part question in regards to the antigen template. So there's a request to do 30 positive, 30 negative, and then that could be retrospectively collected samples, and then 5 positive, prospectively collected samples that are not in transport media. I'm just wondering how FDA is assessing the success of those 5 positives that aren't in transport media. That's one part of the question.
Well, of course we'll look at all the data and make an assessment. Obviously, if they're all negative, that would be a problem, right? So the other thing, just to fill out the response, is getting negatives is really easy. So we'd like to see all the negatives fresh, but you can enrich 25 of those 30 with bank samples. We know that in some cases, freezing the samples releases more target and actually may improve the sensitivity of the assay. So we would definitely like to see that there is no significant drop in sensitivity. But we look at all the data, we make the assessment based on the data that's presented and using the best good science to evaluate.
Okay, and then the second part is the template notes that some can be collected post-authorization. So are the 5 prospective positives included in that post-authorization collection?
Yeah, so if you did 5 fresh and 25 banked, post-market, everything else being equal, an average submission, we would, no outstanding concerns, we would ask for just 25 more fresh samples post-market.
Okay, so what if there are 25 banked and 5 prospective?
So if there are 25 banked and 5 fresh? Yeah. So we want to see complete data ultimately on a minimum of 30 fresh samples. So whatever you don't contribute to that 30 in the pre-market, pre-authorization phase, we would like to see in the post-market study.
Okay, thank you.
So we want to see a total of 30 fresh results, but we are allowing pre-market the enrichment with banked in order to aid in getting particularly point-of-care tests onto the market as soon as possible.
And those 30 fresh results can, sorry to interrupt, but those 30 fresh results can be a mixture of positive and negative, correct?
No. We want to see per application for a point-of-care test, we want to see a minimum of 30 overall fresh positives. So if you only do 5 fresh positives pre-market, we would want to see 5 fresh positives, 25 fresh positives post-market.
Okay, thanks for the clarification.
And to clarify, I believe the template also recommends 30 banked. So we want at least 30 of a complete data set. Whether if your pre-market is using banked, we want 30 banked, even if you have 5 additional fresh on top of that.
You're right, Toby. You're right. Thanks for correcting me. What we want to see is for the banked, that could be frozen direct swabs or it can be frozen in VTM. We want to see a complete set of data for that sample type, that frozen sample type, in order to make our pre-market decision. And we want to make sure with the fresh positive that the test performance is still holding up.
Understood. Okay. Thank you.
Thank you. Our next question is from Sharu Ramkumar. Your line is open.
Hello?
Hello? Hello?
Yes, we can hear you.
Can hear you, yes.
Hi, sorry. So the first part of my question is that if a manufacturer has a legally marketed device and they want to add a COVID-19 diagnostic to that device, I just want to clarify that the information required for the EUA request would be limited to the validation of the assay itself, and we wouldn't need to provide technical information on the device if it's unchanged. And the second question is that if we get such an EUA for a COVID-19 assay on a legally marketed device, can we now apply this assay to additional instruments of the same instrument family under the replacement reagent policy in the same way as a 510K cleared assay would be?
So can you tell me a little bit more details about your assay? Is this a molecular panel you're adding SARS-CoV-2 to, say, other respiratory, or what is this? What is, what are the non-COVID targets on this assay, and what is it doing?
So it's basically looking at a marker for seriousness of COVID-19 infection. So say like a cytokine.
Oh, so okay. Well, we've authorized some IL-6 assays. I'm not sure, other than IL-6, what we've done that haven't been either serology, antigen, or molecular, including panels of respiratory viruses. I think this is specific enough that that you ought to come with, a pre-EUA with what you plan on doing to add that, because it doesn't sound like you're adding a SARS test to a SARS either serology or diagnostic for identification of the virus or exposure to the virus to another serology or direct test. So it sounds like you have inflammatory markers for some other purpose, and you want to add a COVID-19 related inflammatory markers or something to your or apply for the use of your device for COVID-19. And so that is going to be a little bit more unique and will depend on your given panel. In general, when you're adding something to the panel, depending on how that panel works, if the wells are all independent and there's no change in those independent wells, and you're just adding a new well for SARS or wells for SARS, and it has no direct impact on the other wells, then that is going to really make it more streamlined to validate the addition of those new markers, that new marker or markers, because what you're adding doesn't impact the results of the other markers or the analytical results of the other medical. On the other hand, if there's some sort of, everything together, and you're adding SARS probes into already a mix, adding that into the mix could alter the performance of the other markers. So I really recommend that you just write up your idea and send it in as a pre-EUA, and that will likely go to one of our ancillary COVID teams that would review this type of application. It doesn't go in with the regular mix of serology, antigen, or molecular.
Okay, but I think, just to clarify, it's basically there's a device that's, say, already 510K cleared for some other purpose. And my question is now I want to add some kind of cytokine assay to this device. And so, I'm only asking if the information I need to provide is related to the validation of the assay and not the instrument.
Yeah, what I'm saying is, without without unique details to your platform, I can't give you any more specific information today.
Okay. All right. Thanks.
Mm-hmm.
Thank you. Our next question is from Ling Ko. Your line is open.
Hi. I actually think someone earlier mostly answered my question, but maybe I'll just ask a clarifying question about something you said earlier, Tim, and it was around just the comparison of iOS and Android, and specifically just around, different code bases that might be required to basically produce an equivalent app on both platforms. It sounds like from your answer previously that ultimately it's the device equivalency data that is most important to the agency. Is that correct? And essentially, it doesn't really matter how you get there so long as the data approves equivalence, or did we just skip over that part of the question?
Yeah, I don't know how you would carry out the clinical study portion of this for the users, whether you would allow any of their devices to be used or whether you're going to use one specific device in those studies. It might be better to have multiple, whatever the patients have as a device, you use that and you look at the overall performance. Separate from that, we would, because you may only get one result from an Android of this Model X, or whatever, and only one result from an iOS Model 7. So we want to know more data about the comparative performance, if it's actually doing the reading of the test and making the reading results available to the patient. And obviously, as we discussed before, there could be different, they could yield different performance measures based on the fact that the algorithms might be different, the cameras might be different, the resolutions might be different. The background lighting conditions may be less or more sensitive to that. The other thing I thought about is there's all sorts of modes of taking a picture with a smartphone. Essentially portrait and landscape and, night and day, and all those things can, potentially impact the accurate reading of the result. We just want to make sure that whatever, you're going to put into your labeling, that they're all going to be accurate results.
Good. Great. Thanks for the clarification.
Mm-hmm.
Thank you. Our next question is from Elaine Allen. Your line is open.
Hi, thank you. My question is this. If we were to sell a diagnostic test for COVID-19 while the EUA request is still pending FDA review and the EUA were to be rejected, I understand we would need to recall any distributed tests. However, what would be the impact to the patient samples that have already been tested? Are those still valid?
Toby, can I ask you to answer that question?
Yes, absolutely.
And unfortunately, this week again, I'm going to have to leave a bit early. Apologies, but you're in good hands with Toby. Thank you.
Yeah, thank you very much.
Thanks, Tim. And just to make sure that I caught your question, you're asking about if you submit an EUA request concurrently with a 510K?
No. We're submitting an EUA request for a diagnostic for COVID-19.
Mm-hmm.
It's, we're under the notification, so we're distributing the test. But should the EUA request be denied or rejected? Now we've distributed tests, and of course I understand we'd have to recall any distributed tests, but what would be the impact of the patient samples that have already been tested? Are they still considered to be valid?
Got it. I'm glad I asked for that clarification since I had missed that nuance there. That is something that we would work with you as we concluded our review and went to a denial. We would work with you depending on the impact of the reported results, whether we expected you to do a communication to all your clients or work to send out corrected test results or invalidated test results or anything like that. It would depend on the specific circumstances, and we would work directly with you on that.
Okay, so So if I understand, just for clarification, there would be some sort of a rejection agreement, and that within that rejection agreement, it would be outlined what would be necessary, whether to contact customers that have performed testing and what we may need to do as a result of that.
Right. It wouldn't necessarily be part of the denial, but it would be something that we would work with you on after a denial decision was made.
Okay. And so it depends, right?
Right. Exactly. Always the best answer, right?
Well, the project team's going to love hearing that. Thank you very much. I appreciate it, Toby.
No problem.
Thank you. Our next question is from Esther Yang. Your line is open.
Thank you. My question is simple. Back to the new guidance document. I'm not sure how prescriptive that document might be, but as it relates to mutant genotype detection or confirmation, is there a minimum number of samples that are required to make that call? Hello?
Sorry, forgot to take myself off mute there. So I'm trying to think through the specifics of the guidance, and I don't believe that it gets into that level of detail, but that is certainly something that you could come in with a proposal regarding.
So with the pre-stop? Okay.
Yes.
Yep. And related question in the sample stability testing, are you required to use just the natural samples only, or can we supplement some with the contrived samples?
Sorry, that was regarding sample stability?
Correct. Stability testing.
I would have to look at the template to see what is recommended there. Let me see if I can find that very quickly, and if not, I will ask you to send that in. Okay, I believe that you can use inactivated virus for sample stability.
Thank you.
Yep.
Thank you. Our next question is from Laura Ferguson. Your line is open.
Hello. Thanks for taking my call. I wanted to know what the cutoff is, or what order of magnitude was being referred to with regard to laboratory tests being considered high priority for, I believe the term Tim used was significantly high throughput. What level of throughput is that?
Thanks for that question. I believe Tim addressed this a little bit last week on the call as well. We don't have a firm number that we've put out, because this does change a little bit depending on other details, such as submissions in-house and as the pandemic progresses. So if you do have a specific question for your test and wanting to know whether it would be considered high throughput or significantly high throughput, you can send that in through the mailbox and we can take a look.
Okay, thank you.
Mm-hmm.
Thank you. Our next question is from Shannon Clark. Your line is open.
Hello, this is Shannon Clark. So my question is in relation to, with UserWise, this question is in relation to antigen point-of-care studies. The antigen template notes that the point-of-care study should be run in the United States. And to obtain point-of-care use of an antigen test kit, can we run the point-of-care study in Brazil with 6 untrained healthcare professionals? If we submit a justification of equivalency between the Brazilian healthcare providers and the U.S. healthcare providers, and as part of this, can we translate the point-of-care instructions into Portuguese via a certified translator and still use a study such as this?
So we definitely recommend that these studies be conducted in the U.S. Because we have seen differences between populations in in the U.S. and other countries, specifically with how practice of medicine and how healthcare providers are trained, et cetera. If you have a proposal specific to your situation that you'd like us to consider, we always consider alternate proposals, and you could send that in. But we highly recommend that you do that in advance of starting any study in case we do not agree with the proposal.
And can I submit a study synopsis rather than the full protocol in order to get clarity sooner, or do you require a full clinical evaluation protocol?
You can submit the synopsis. It is quite likely that we would request additional details as we take a look, though.
Okay. Thank you so much.
Sure.
Thank you. Our next question is from Sue. Your line is open.
Hi, Toby. Thanks for taking my question. I think part of this was already answered, and I don't want to belabor this. It was around the antigen performance and the 5 prospective samples. I just want to know if you could clarify, so that the samples could be collected retrospectively or prospectively, but then in addition, the submission would need to include those 5 prospective samples without transport media, and then while you're continuing to get the 30 positive throughout. And then I'm just looking at a recent IFU that was just published, I think, this week, where it's intended for fresh samples, but the clinical performance is all on bank samples, all on frozen samples. So I was just curious if you could talk a little bit about that.
Right. So we would want to see a complete data set of 30 specimens. Without knowing exactly which test you're referring to, I can't comment on the IFU, but we would want to see, a complete data set of 30, and then if you have additional fresh, that would be on top of the 30, and we would ask for the rest of the 30 fresh to be in the post-authorization.
Okay. Okay, great. Thank you.
Thank you. Our next question is from Debs Payne. Your line is open.
Hi. Thank you for taking my call. This is Debs Payne, CPDP Consulting, and we're interested in exploring validating a mouthwash collection, alternative collection type on our to amend our submitted EUA that is using nasal swabs. I had reached out to the templates email, and they had told me to contact my primary reviewer, but it's been 7 months and we don't have a primary reviewer for EUA. So I'm seeking guidance on what that might look like, what that validation might look like.
Okay, so I would start by recommending that you take a look at the alternate specimen type validation information in the template. That would be a good starting point. And then if that does not seem like it is applicable to your situation, you can submit the questions again through the mailbox, and just flag that you don't have a lead reviewer, and so you would like to get some input. And if you want to direct that or ask that they include me on that email, you can do that, and they'll flag it for me.
And what is your email address?
Or you can just send it to the EUA mailbox, and they'll add it. They'll forward it to me.
Okay, because I think that you have an alternate one for saliva, but the mouthwash is quite different.
Right. That's why I suggested taking a look at that alternate specimen, which you're right, is for saliva, to see if that information is applicable. And then if you have additional questions on top of that, you can send them in.
Thank you. Y'all are doing a good job. I'm hoping to get a response from a reviewer one day, but patience is a virtue.
Thank you for understanding.
Okay, thank you.
Sure.
Thank you. Our next question is from Nisha Lee. Your line is open.
Hi, thank you for taking my call. My question is regarding the variant testing, and our company is developing a POC antigen test. we've looked to places like BEI and have been successful in obtaining inactivated virus. So my question is it acceptable to FDA that we run an in silico analysis on the protein alignments of the nuclear protein from each variant and map this against our antibody epitopes? So the idea is that if our epitopes do not map near these regions, then we would have a very low risk of seeing a decrease in our performance.
And I'm sorry, what type of test is this for?
Oh, this is for a POC antigen test.
For antigen, okay. I wasn't sure if it was antigen or serology there. That's something that it would probably be best if you can send in your proposed plan, and we can take a look at it and get you some feedback.
Okay, thank you.
Sure.
Thank you. Thank you so much, Toby. I believe you wanted to make a clarification. That was, in fact, our last question, so you can go ahead and clarify.
Great. Thank you, Irene. I just wanted to jump back to a question that we had earlier in the call about enrichment due to reduced positivity rates. Tim did address this a little bit with talking about the potential downfalls of enrichment, but we just wanted to clarify that enriching your study with known positives can cause bias. So we would expect your study design to include a plan for addressing that bias. So we just wanted to jump back to that topic and make that clarification in case that wasn't clear in the previous remarks. And that's all for today, I think. I'll turn it back over to you, Irene.
Thank you so much. Thank you. This is Irene Ahir. We appreciate your participation and thoughtful questions during today's town hall. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Friday, March 5th. If you have additional questions about today's presentation, please email cdrh-templates@fda.hhs.gov. As we continue to hold these virtual town halls, we would appreciate your feedback. Following the conclusion of today's virtual town